the hands of late Padma Bhushan Dr. Vasantrao Patil. He was the great freedom fighter uh, and 3 times Chief Minister of Maharashtra, and he was ex-governor of Rajasthan. So he's a great personality, and he had mission to open up this engineering and technological education to all rural masses so that we can gear up the industrial development. And that is really, therefore, the Maharashtra today, if you look at the Maharashtra, uh, it is advanced state, one of the advanced states in India. And because of this industrial culture which has grown over the last many years, and Dr. Vasantrao Patil was one of the key personality in looking this height. And this institute is developed, started by him in BUDHGAON, Sangli. Sangli was his hometown, and therefore he has started this institute. Since 83, we have this instrumentation engineering branch in our institute. Along with instrumentation, we have other branches like civil, mechanical, chemical, electrical engineering, electronics, and electronics and telecommunication in computer science. So all we have total 8 branches, and we have about 630 intake every year. And there are about 2,500 plus students in the campus studying every year. And along with this engineering education degree, we have also polytechnic, wherein about 1,000 students are studying in the campus. So total about 3,500 students are studying in the campus every year. And this is a big institute that way. And most of the students are from rural area nearby Sangli. The very few are from metropolitan cities like Mumbai, Delhi, and OMS, outside Maharashtra state. So our challenge is to groom these rural students and convert them into very good engineers, useful for society, useful for nation, to rebuild India. And therefore, we are arranging all these webinars on various issues related to the societal development. So we are involving our alumni as well as the experts like you who have worked in the field, respective fields, so that the students get an idea and they can decide their own path. They can think about their career. They'll get some good inputs so that their thought process can be, you can say, given some direction. And that is our basic motto. And we're working on these lines. We're working on innovation, uh, culture in our institute, in our campus, and we are spreading this awareness amongst all students, including our institute as well as other institutes also. So we are conducting many such workshops for our students as well as for students studying in other colleges, may it be diploma, may it be science and science, BSc, MSc, or in other engineering institutes. So due to this pandemic, again, we're not accustomed to such kind of online teaching learning process, but because of this pandemic, we are disrupted, and we are sitting at home and trying to utilize this time for getting some additional knowledge apart from the curriculum. And that's why we have called you here, and uh, you have accepted our request. Dr. Nandikishore is coordinating this event, and he has called many such personalities nationally, internationally, and therefore this webinar is one of the different kind of webinar wherein we have included variety of topics. And I am very glad to know that you are also here and you are going to talk about the PCR. So really, we are all worried about. We don't know what is PCR, what is corona, and how it is getting transmitted. How does it attacks our systems? And it is really a hot topic today. And therefore, I am very much glad to know that you are going to talk on this, and we can take ahead our association future endeavors also. So I welcome you. Will not take much time. I welcome you once again on behalf of Pvpit and our management, Chairman Honorable Vishal Dada Patil and our trustee Honorable Amit Dada Patil. We are always supportive for such kind of activities, and they want to go ahead in these lines itself. And therefore, once again I welcome you, and I thank all my institute department, faculty members, coordinator Dr. Nandikishore, for giving opportunity for me to interact with you. Thank you.
Thank you so much,
Yes.
Professor.
Yes, so thank you, sir. Before Dr. Dhokane sir, you actually speak. Let me just first speak some other words. So number one, I wanted to mention, the scope of this talk today is it's a twofold. Number one is because of the PCR, which is, as our principal mentioned, that it's a hot topic because of the coronavirus. So that is actually one fold. The other fold is because we hail from instrumentation engineering department, and I believe that the end users like you and the customers like you actually know the in and out about the instruments, and probably a lot of issues that instruments have, and then if you can advise what could be an instrument actually be like, from ergonomics point of view, from usage point of view. So because we are from instrumentation engineering department, so our students and faculties can understand those kind of problems, and then maybe we can try to re-engineer or rework on in building instruments which are more with less problems, or more easy and ergonomically better. So that is the other side. The other side, actually, another I wanted to mention, so if you can also I'm sure that you have actually, you're going to bring that forward, but if you can mention about what are the different instruments that are used in the lab apart from PCR, and also if you can actually mention what are the make, model, companies, and the prices of those different instruments, like scientific analytical instruments, so our students can also get aware about that, and probably they can actually start thinking about another career in the field of instrumentation engineering. Because right now many students, what they think is instrumentation is more or less something right with respect to automation, but scientific world, India, we are actually leading on that front. We are probably on the third or the fourth rank right now across the globe. So we also we are doing a lot of scientific research, and we are actually having a lot of instruments to do a lot of work. And then I just want our students to be aware of that, and so they can actually join industries that do such kind of work, and they can actually probably have access to it. So that is the third reason, why I was trying to do this session. So thank you so much again, Principal Sir, for interacting with my friend Dr. Dhokane Sir, our guest of today's speaker. And now I would like to request our HOD to start the session. Thank you, sir. Thank you so much.
Thank you, Dr. Dhokane sir, for presenting here and giving us the valuable time. And I will request now the Smita Patel madam to give the introduction of the today's speaker.
Thank you, sir. Hello. Good morning, all of you. Today is the last day of student development program on future trends and career opportunity in Industry 4.0 in India and abroad. Today with us, Dr. Dhanajai Dhokane sir. First, I welcome you, sir, on behalf of Padma Bhushan Vasantrao Dada Patil Institute of Technology and Instrumentation Department. I also welcome to our principal, Dr. D.V. Ghevde, sir, all participants, all deans, HOD and faculty of PVPIT. It's my great pleasure to introduce today's speaker, Dr. Dhanajai Dhokane, sir. Sir completed his PhD in plant science from McGill University, Canada. Currently, he is working as a research scientist, synthetic biology in Reliance Industry Limited, Navi Mumbai. He is affiliated with several professional bodies such as the Canadian Association for Plant Biology, Biotechnology, Canadian Society for Plant Biologists, Canadian Society. He received total 10 awards related to the plant biotechnology, plant biotech, and plant science. Today's his talk will the know more about the PCR instrumentation. Hope this will be the helpful to all of us. Once again, I welcome you, sir, and I request you, sir, to start the today's session.
So good morning, all. Am I audible to all?
Yeah, yes.
Yes,
Yes,
you
sir.
are.
Okay, perfect. So good morning, all, once again. I would like to extend my thanks to Dr. Nandakishore, Dr. Gevde, and all the organizational committee who has organized this webinar and have given me opportunity to interact with your students. And you guys are eager to know about different instruments, what are being used in labs, what are its usage, how they are used, how you can make improvements in these instruments and can serve the community better. So I would like to thank everybody who has devoted their time in developing this program. So I will start with the outline of today's talk will be PCR and its basic, how this PCR reaction, what is actually PCR reaction, why it is used, what is the history of this PCR, what are the components of PCR reaction, why actually we do PCR reaction, what are the conditions for PCR reaction. Then I will move towards basic requirement of PCR, then towards your topic of interest, basically instrumentation, then vendors who supply these instruments and approximate cost of each instrument, and then the other uh, lab equipments those are used in labs. So to start with, actually what is PCR? So PCR is an basically a reaction that takes place in a PCR machine or a thermocycler, or it's called a DNA amplifier. Now, to just simplify what PCR means and what PCR is. So if a human genome has 40,000 genes, and if you want to isolate gene number B, so how you will do that? Because in the genome you have one or maximum two, or maybe if it is a multi-gene family, you may have more than two genes. So if you want to isolate one single gene, it's not possible to get enough product of that gene after doing PCR. So what is PCR is, out from a small segment, if you see the picture here, you say the template DNA. This is your gene of interest, which is marked in red. And then when you want to, if you want to make multiple copies, you use this reaction called PCR reaction, that is polymerase chain reaction. Basically, herein you use an enzyme and a combination of different temperature cycles, wherein you make more copies, I would say million copies of a single fragment. When you have a sufficient amount of amplified product or more product you have, then you can easily purify your product and you can go for transgenics. Maybe you have heard about BT cotton, BT maize. What exactly is a gene from different bacteria is, here multiple copies of these genes are done, and then you purify the gene, and then this is introduced into a new organism that is called transgenic. So what exactly is PCR? The technique that makes more copies of a DNA segment or a gene. But there is a limitation of this PCR reaction or a PCR volume, I could say. It makes only copies of small fragments. It can't make copies of a genome. So if you want to amplify something that is 1 or 2 kb in size, or 5 kb or 10 kb maximum in size, you can do that with PCR. But you can't completely amplify the genome or make multiple copies of your genome. And this is an in vitro technique in which you can make in that you can do it in a PCR tube, that is small plastic Eppendorf tube. So if you have any question in between, you can stop me and you can go ahead. So when we need more copies of a gene to do some downstream applications, you need to do a PCR reaction so that you have multiple copies of a single gene, and then you can clone it and express in different organisms. So what is the history of when was this PCR developed? So in 1983, Dr. Carey Mullis had developed this PCR reaction, and then we had a first publication on PCR in 1985 by Citrus Corporation. And then, you know, I told this reaction consists of an enzyme along with changes in the temperature that makes multiple copies of a single gene. And Perkin Elmer was the company that introduced automated PCR machine in 1988. And from
Hello.
then,
Hello, sir.
yeah, go ahead.
How do you copy a gene? It is the exact.
So basically, this is an exact replica of a gene. So if you want to extract a gene number B from the genome, you need to design, that is in technical term, that is called primers. I would say signatures for you to understand. Those signature binds on your gene of interest on both the ends, and that those primers extend the gene of interest and make multiple copies. Is that clear?
Yeah, but copying means actually you're copying a gene means you're developing a new gene.
No, copying means we are making more copies.
That is what
It's
actually
like a Xerox machine. I will just explain you how it happens. Suppose if you want to make a copy of gene number 2 in the genome. So what you do is, we know the sequence of gene 2 in the genome. Then we design a set of signatures that will sit on our gene of interest, that is gene number 2 in the genome. And using this enzyme, and you have DNTPs, that is dinucleotides, that are incorporated on these strands, and with the cycling, temperature cycling, it helps in the extension of the strand. I will come into the details in the upcoming slides.
okay.
So now if you see what are the components of a PCR reaction, so you need to have a DNA template. DNA template is the starting material on which you make multiple copies. Suppose if you see this DNA template, you design something called signature, which is called primers. These primers will attach to your gene of interest because this will have sequence similarity. With sequence similarity, these primers sit on your DNA template. This is an enzyme called DNA polymerase. These enzymes incorporate these bases which are complementary to your DNA template. So basically, you guys might know ATGC are 4 DNTPs or 4 bases or 4 nucleotides that makes our DNA. So these DNTPs are being incorporated. Suppose this is our gene of interest. Your primers get attached to both the ends based on our designing. Then this polymerase starts incorporating the bases on the bottom strand as well as the upper strand. So in the first reaction, if you have one gene in the genome, in one cycle, it makes 2 copies. So in order of 2, it will make. So in the second reaction, you will have 4. Then in third reaction, you will have 8. Then you will have 64. So it's like magnitude of 2. So a PCR reaction consists of a DNA template, which is a starting material on which your signatures they bind. And then you have your DNA polymerase, your DNTPs, your buffer that is necessary for activating your DNA polymerase, and the cofactors that are essential for DNA polymerase activity. You put all these reagents together in this plastic Eppendorf tube, and these tubes goes into a PCR machine. And that way this PCR reaction works. So what is the purpose of PCR? So as I told, to amplify a lot of double-stranded DNA molecules or fragment with same identical size and sequence by enzymatic method and cycling condition. Suppose if you see this is your double-stranded a gene of interest, in you denature this fragment into single strands, and these single strands, your DNA polymerase, your DNTPs, they act on and make multiple copies of your gene of interest. So once you have multiple copies of your gene of interest, then it helps in downstream processing. So what we guys do is, once you have multiple copies, then we extract that gene of interest, clone that in a vector, and then transform it into our host of interest, maybe Arabidopsis, maybe cotton, maybe wheat, or whatever species you want to transform. That becomes transgenic approach. You can also sequence the gene to know what exactly the bases, that is ATGC, is present in your gene of interest. So with this, you can do sequencing, you can do cloning, you can transform this gene of interest, you can do molecular diagnostic, you can identify mutations. Similarly, in this way, if you know the sequence of coronavirus, you can design 2 signature primers. Those will sit on the gene that comes from coronavirus. And then you keep this mixture in a PCR machine that makes multiple copies of this coronavirus gene. And then you load this reaction onto agar gel, where you can clearly see if your patient is positive, you will see a band. And if your patient does not carry any gene from coronavirus, it will not show any band on the agar gel. So this is how polymerase chain reaction is being used for coronavirus detection or screening. So if you come to conditions for PCR, so PCR reaction comprises of 3 steps. So your DNA, as everybody knows, your DNA is double stranded. When you want to make multiple copies of your gene of interest, you need to denature your DNA into 2 strands, that is strand 1 and strand 2. And then you have an annealing. You see here your primer, which is sitting at the end, and you have your forward primer, which sits on the other end. Primers are nothing but the signature which are exactly matched to your gene of interest or your fragment of interest or the gene that you want to amplify. Then using, see, if you see the temperature range here, you see denature occurs at 94 to 95 degrees Celsius. If you see the primer annealing at which your signature or your identical fragments of your gene of interest, they bind to your fragment at 50 to 56 degrees. And using this DNTPs and polymerase, you get your fragment extended. So you have same exact replica of your fragment and in multiples. Third is the extension step, where your double stranded molecules are formed and you have a complete amplicon, that is called amplicon or a fragment. So what are the basic requirements of a PCR reaction? Suppose if you want to identify or see the sequence of a coronavirus gene. So what you should know the sequence of that gene. That is very much prerequisite for designing of your primers or signatures, I would say. Then your signatures that recognize your fragment and help in making more copies. Then, as I mentioned earlier, you need a thermostable polymerase that can withstand a temperature range of about 100 degrees Celsius, because your denaturation of your double stranded DNA takes place at 95 to 94 degrees Celsius. So we need a polymerase that can withstand this temperature. And then you need a thermocycler machine or a thermocycler or a PCR machine or a DNA amplifier that can be programmed to carry out heating and cooling of samples over a number of cycles. If you see the basic requirement for PCR, as I mentioned in the previous slide, you need a template DNA or a fragment on which you need to make multiple copies, or you need to make more copies of that. Then you need primers. Those are nothing but a small fragments of DNA. Those sits on your template and they help in making more copies. And these are all the reagents that are needed for a PCR reaction. All this mix goes into a PCR tube, and these PCR tubes go into a PCR machine. So how does this PCR works? So if you now, I think you all are aware about what are the components of PCR. So basically, I would say your DNA samples or your throat swabs are your starting material. Then you design a small set of primers, which are signatures. They specifically bind to your gene of interest. Suppose if you want to identify a coronavirus gene, you have like a throat samples, and then you design primers or a signature. Those will only sit on coronavirus gene, and others, and these primers will not sit on any gene from the human. So basically you can only amplify a gene from coronavirus. Then these are the nucleotides that are being used for incorporation and making double stranded fragments. This is a PCR tube wherein you put in all the reagents, and this tube goes into a thermocycler, and based on using thermocycler or a thermocycler, the PCR reaction consists of 3 steps. That is denaturation, wherein the double stranded fragments get separated into 2 single strands. at 94 to 95 degrees Celsius. And then these two strands, strand 1 and strand 2, wherein your primers they bind, and you see you have like 4 copies of a fragment in a one reaction. So the 3 major steps are the denaturation of your double stranded DNA, then annealing of your signature to your gene of interest so that it sits on your gene of interest and make multiple copies for you. And third is the extension. Once your primer sits, it extends your fragments to the complete length of interest. Suppose if your gene of interest is 700 base pair or 1000 base pair, it makes 1000 base pair or 700 base pairs. So coming to a PCR machine. So if you see in the image, the PCR machine looks something like this. So you have an heating block upside. If you see the pointer, this is an heating block. You have an display here, and you have a keyboard here to set up an protocol. So basically using this keypad, we set up a protocol using different temperature range. Then we can adjust whether if you want to incubate some samples at 37 degrees, you can incubate those samples. If you want to incubate samples at 60 degrees, you can do. So basically, if you have seen, you need 94 degrees Celsius, then you need something around 60 degrees Celsius for your primers to bind. And then you need an extension of 70 degrees Celsius where your fragments, they completely extend to their maximum length. So this is and this is an upper lead. So basically here your PCR vials or PCR reaction sits in, and then you close this lid, you set your protocol saying I need this temperature for this cycle and how many cycles you need based on your biological output or what is your downstream purpose. Based on that, you put number of cycles. Usually in a routine molecular biology labs, we put around 30 cycles. So we have more than thousands copies of a gene. So PCR machine is also called a thermal cycler or a DNA amplifier, in which you make exact multiple copies of your gene of interest. It's like a Xerox machine. If you put one paper, it will make multiple copies of your paper of interest. So it regulates temperature during cycling program. So basically this is an instrument in which you can regulate the temperature regime. So from 0 degrees Celsius to 99 degrees Celsius, you can play around with all the temperatures. So heating and cooling are the two steps that are very important in a PCR machine. If you have a heating block wherein the temperature is being regulated, you can do your PCR. And why cooling is necessary? Even if you keep a PCR reaction at like 5 o'clock in the evening, and if you go home, you can keep the machine running, and the machine will attain 4 degrees like your freezer, and these samples will be stored at 4 degrees in the PCR machine itself. Next day morning when you go to work, you can take out your PCR reaction and then load on gel and see whether you have your fragment of interest amplified. So all the components of a PCR are placed in a small tube called PCR tube, and then the tube is placed in the machine. So exactly this, you have a machines of, you can place 1 tube, 2 tubes, 96 tubes, 384 tubes. There are different versions of those machines wherein you can put like you can do about 384 samples at a time. If you see what are the functions that are embedded in a PCR machine. So PCR or a thermocycler is a machine where you can set different temperatures ranging from 4 degrees to 99 degrees. So basically 4 degrees act as your freezer, wherein you can store your samples once your PCR reaction is done. In simple words, PCR is a machine where a temperature can be set in steps to carry out a reaction to have a final product. Final product is nothing but you have a copy, more copies of your fragment of interest. You can also incubate your reaction at different temperatures. So as I told you, in this machine, you can set temperature from 4 degrees to 99 degrees. So you can also incubate in molecular biology. There are other techniques also wherein you need incubation. So incubation at 40 degrees, incubation at 60 degrees. So you can also use PCR machine for incubation for different time points. You can also store your products at 4 degrees, like stored in refrigerator once the reaction is done. So You can also use PCR to store your PCR reaction overnight once the reaction is done, so that next it won't get denatured during the night, and you will lose your time. So basically you can save the time, and you can load your samples next day on the gel and see if you have your fragment of interest. So now coming to the advantages of PCR. So why this machine has become so much popular? What are the advantages? Why every lab wants a PCR machine? Why every molecular lab, biology labs need this machine? So if you have one copy to start with, you can make multiple millions and trillions copies of a signal fragment. So this machine is very accurate and precise, and you can make multiple copies starting with a small starting material. Second thing, it's automated. Now it's fully automated. Now you have a tabletop PCR machines wherein you can just automate your protocol. No need to sit near the machine. You just have your tea, lunch, and come back within an hour and a half. Your PCR reaction is done, and then you just go for visualization of your results. So, and this machine produce very reproducible results. So if you do this reaction multiple times, you get the same results. So as I told, within hour and a half or two, you can get your results. So this is very fast and very quick and very precise in size determination. So if you want to identify the size of a gene, you can design 2 signatures, primers, and then you can do this PCR reaction, and you can also determine the size of your gene of interest. You can also detect mutations. So now if you know these hereditary diseases are caused by some mutations in your genes. So genes causing those diseases. So using this PCR also, you can amplify your target gene that cause those disease. Then you can send it for sequencing, and then you can compare the gene sequence of a healthy patient with the gene sequence of a disease patient. If you see some sort of variation in the nucleotide, that means something is wrong in the disease patient. And then you need to work with the disease patient to correct that gene sequence to make or regain that patient to a normal condition. So what are the applications of this PCR? So PCR.
Excuse me, Dr. Dhokane. Actually, it's Dr. Nandkishore here. So I just wanted to interrupt, for just a few seconds. So my question was, and correct me if I'm wrong. So my question is, so this PCR instrument, it is actually it has 2 sides. Number one is in your field, where you probably do breeding, you work in breeding, right?
Yeah, molecular biology and breeding.
Yeah, molecular biology. So in that the use is actually if you want to actually incorporate some kind of a new kind of DNA into a plant, which will actually probably, let's say it wants to improve its productivity, or maybe it wants to improve the number of fruits actually bearing capacity, or maybe the nutrition quality. So you actually identify certain gene, and then you actually run it through the PCR, and then at the end you have to detect if what you actually wanted is it really amplified. So there is some kind of a detector, right? You said something agar gel or something.
Yeah, agar gel.
Yeah, agar gel. So if you can also mention something more about that. So that is the detection part. So if the PCR reaction has been a success, you can only know you can only know when you detect something, right?
Yeah. So I'll talk on that front episode.
Yeah. And then from the corona perspective, what we are doing, we are amplifying that because we want to detect it. So if that gene is present, if the coronavirus gene is present in the swab, then we amplify it, and because the detection needs some kind of a signal, which is if there are only 2 or 3 genes, it will not be able to detect. So there has to be some kind of a density of that genes. Only then there will be some kind of a detection, because there is some detection capacity or some kind of a resolution on the detection part. So I think that's why we are actually doing the amplification, right? So from the corona perspective, so that probably I wanted to actually probably clear it out, because that's what I actually probably thought, because probably you didn't mention about that. So thank you.
Okay, Nandakishore. So actually what we do for corona screening is, so we collect swabs from like a patient which we see they are suspects. And then what we do is we extract a DNA or an RNA from those patients. So it can PCR can be done on both the samples. So what is done is when you have a DNA sample, then as I mentioned, those primers are signatures. Those will only sit if you have a coronavirus gene present in your swab. If you are a healthy patient and if you don't have any coronavirus infection or a virus sitting in your swab, you won't be your primers won't be sitting on any gene that comes from human genome. So those primers will only sit on the coronavirus gene. Then we will put the swab along with your primers and other components of a PCR in a tube. That tubes go in a thermal cycler. Then it makes multiple copies. And then once this reaction is done, we take 10 microliter volume out of that and we load on agar gel. This is a detection method, as you clearly said, on which if you have a coronavirus gene, then only that gene will be amplified and you can see a band on your agar gel. If you don't have any coronavirus gene present, it won't be amplified and you won't see anything on the gel. You will see a blank gel. That means you are not infected with coronavirus. You don't have any gene that comes from coronavirus. And in the patient, those they carry coronavirus or they are infected with coronavirus, they will have the particles of this virus. Then from that DNA will be extracted. Then your primers will sit clearly on the coronavirus gene and it will make multiple copies and those copies will be detected on the gel. That's how we say he is a positive patient or she is a positive patient and he or she is a negative patient. Is that clear?
Yes, thank you. And how is it detected? Like, is it like visual detection or is there like some kind of a microscopic camera detection to actually detect the band?
So basically, we detect it under a UV machine. You can see that on your computer screen. You just place the gel. Gel is nothing but it's made from agar. No, it's made from agarose. You just melt agarose, then cool it, and then in the agarose gel you have lens or wells, I will say. In those wells, you just pipette out to like 10 microliters and run it. As DNA is negative charge, it runs from negative to positive, and then when it runs, you see something on the gel. I will also show you how the gel looks like. It shows like a thin or a thicker band on the gel. Suppose if the coronavirus gene is 700 base pair, we have a marker along with the samples running, and your gene of interest band will clearly be seen at 700 base pair, very close to your marker lane. This is like we add something that intercalates between the DNA bases, and under UV light it gives fluorescence. Is that clear, Nanda Kishore, or anything?
No, it is clear. Thank you. Sorry for interrupting you. You can continue with your next slide, sir.
So if you see the applications of PCR, it has wide applications starting from molecular identification, sequencing, and genetic engineering. As Nanda Kishore clearly said, what we are doing is we are trying to do trait improvement. Trait improvement means you have wild varieties and you have cultivated varieties. So if you see the wild varieties or wild species are very resistant to or tolerant to some biotic and abiotic stresses. Suppose if you see jawar is very tolerant, it can withstand water stress. So what we do is we identify genes that are responsible for like drought tolerance or that can withstand this water stress. We amplify those genes using PCR. We make multiple copies of those genes. Then we clone that in a vector that is called a delivery vector. And that gene is then introduced into, say, suppose wheat to make it more drought tolerant or it can withstand water stress. This is how PCR is being used for making multiple copies. And then these copies are cloned into a vector that is called a plasmid vector, and then you transform that into your host of interest. Your host of interest may be tomato, potato, any plant species, any microbes, you have the same routine, same techniques used in all the, let it be humans also. So basically, first you identify a gene, then you amplify or make multiple copies of that gene, then you put that fragment into a vector that will act as a vehicle to transport that gene into your host of interest. Your host of interest may be anything that you want to improve. So if you come under molecular identification stream, it can be used for DNA fingerprinting. You can classify the organisms based on what genes are there. You have signature genes. Then you can do genotyping. So what bands, whether you have heterozygous, homozygous. Then you can do prenatal diagnosis. You can do mutational screening. You can do drug discovery, then genetic matching. Then you detect pathogens like coronavirus. And if you come to sequencing part, you can do bioinformatics and you can do genome cloning. As you know, the Human Genome Project, all the genes were amplified and they were put together to form a Human Genome Project. Then you can, using PCR machine, you can incorporate changes into your gene of interest. Then you can modulate gene expression studies. So these are all the applications of a PCR. So now coming to what type of PCR instruments are available in market. So as I mentioned in the previous slide, you have machines with 96 well plates. So see, if you see this plate, it has like 96 wells, wherein your 96 samples can sit here. And if you close the lid, you set your cycling protocol, and then you hit the run button, you wait for 2 hours, and your PCR reaction is done. Then you load these samples on agar gel to see your band of interest. So then you have machines with 384 wells. So if this is the machine with 384 wells, where you can use a plate wherein you have 384 wells, and you just put that plate into the block, and you again set up your protocol, hit run button, and it will start amplifying, making more fragments of your gene of interest. And then you detect, as I said, using agar gel electrophoresis. Then you have these kind of machines where you have two individual blocks. You can set one kind of reactions in this block and the other kind of reactions in this block, because the annealing temperature is the temperature at which your primers, they bind to your fragment of interest. So then if you see this digital display, you have, if you see file, you have saved protocols, then you have incubation, and these are miscellaneous folders. So what you do is you hit this button and set a new protocol starting with denaturation, annealing, extension. So this is also a similar machine, but here you have instead of digital display or touchpad, you have a keypad over here, where using which you can set up your protocol. And this display shows you how much time is left for your PCR reaction. And this is a single thermal cycler from Biorad, wherein you have a digital display and a touchpad, wherein you can preset your protocol, and according to your gene of interest, your primers, its annealing temperature at which the primers bind to your fragment, and then you just hit the run button, it will start making multiple copies. When you come back after hour and a half, you will see that your PCR reaction has been done, and then you just load your sample onto agar gel and see whether you have your band of interest. So what are the features that end user look for? So if you see the different machines, so the Peltier effect technology, then upgrade to real-time PCR. So there are different types of PCR. So what we use in coronavirus is RT-PCR, where you have cDNA as a template. Most of the times you have a DNA as a template. Then whether the interchangeable sample blocks, as I said, in this you can interchange the temperature, and you can use this block. You can use one block for incubation, one block for cycling temperatures. Then what number of wells are present? See, if you see 96 well plate, 384 plate, 48 dual plate. Suppose instead of 96, you have 48 well, 2 blocks. And then you have machines with 384 wells, and you have only single 96 well plate machines. Then whether you can set the gradient temperature at this cycle, I need couple of temperatures, where if you can do that, that is the gradient capability. Then graphical user interface, as I shown in this machine, you can see the graphical user interface. You can see which cycle is running, what is the temperature, how much time is left for your PCR reaction, and then fast PCR protocol templates. So if you have any thing built in your machine that will help you to preset your protocols. Then choice of temperature control mode, whether you have automatic temperature control, how it drop downs, how it rises up, to what degrees it can goes, what is the maximum and minimum limit of temperatures. So power failure restore, whether it can restore once the power is failed, and once you switch it on, whether it can restore your running protocol. So programmable ramp rate, so whether you can ramp your temperatures. For certain amplification, we need to ramp certain steps at different temperatures. Then networking capability, adjustable heat lead, then full skirted plate. These are all the features that an end user like me, they look for. The more sophisticated, the more handy machine we prefer, because you need not to do anything. You just keep your PCR tubes in the machine, you just uh, preset your protocol and just hit the run button. The PCR machine will start rising the temperature at initial your denaturation step. Then it will start, the temperature will start dropping down. Then your signature or the primers will start sitting on your fragment of interest, and then again it will go a little higher to 72 degrees, wherein your fragments will get extended to the size that is of interest. Suppose if your size is like 1 kb or 700 or 500 base pair, the temperature and the timing depends on your size of interest. So these are all the features what an end user look for. So which companies makes these PCR machines? So who are the like vendors providing these PCR machines? And uh, few of them I have listed here are like Biorad, then Roche, then Thermo Fisher Scientific, then Perk and Elmer, and then Agilent Technologies. Like if students are interested to attend internships or get some hands-on these instruments, you can look for internship courses or internship at these institutes. And these are, I would honestly tell you, these guys are the ones who are running science. They are much ahead than us. These guys, they make bring in new modifications, new automations, mm, and they always look or talk to people who work in labs to figure out what is actually needed, what upgradation is needed in this machine to make it more user friendly. So these are all big players, and these are all multinational companies. Those make these PCR machines, and the range of PCR machines in Indian currency, it goes from $25,000 to around 1 lakh. And if you want to buy RT-PCR machines, they are around 2.5 lakhs, 5 lakhs. So it depends on which company, what is the capacity that you can put in. Suppose 384 well machine goes much costlier than 96 well played. If you have a small lab, you can even go for 48 well machine. And then if you see what features you want to go for, again the pricing varies with the features. The more the feature, the more the costing. And Thermo Fisher and Biorad are like the leading companies in this technology, like for providing PCR machines. And as I mentioned in my second slide, Perk and Elmer was the first company to introduce the PCR machine in 1988. So these are like key players or vendors, those provide PCR instruments. And students can also look for like internship in these companies, because these companies, they produce lot of lab instruments ranging for PCR machine, agarogel electrophoresis, and laminar workflow. I will come to other instruments in later part of my presentation. So, oh no, here I'm coming. So what are other instruments that are being used in labs? So if you see, this is a robotic system wherein this is called a colony picker. You just place your petri dish in the center, and you command your instrument such that it only picks blue colonies, or it picks only white colonies, or it picks only bigger colonies. You give whatever instructions are being needed for a robot or this automatic colony picker to work. And then this will pick your colony of interest, and this colony will go into your PCR 96 or 384 well plate, and then you can add your PCR components and just keep this PCR plate into a PCR machine, and you will have your gene of interest amplified. So these are, see, it takes a single plate one by one, and it keeps it here. This is the picker which picks, and then you have a sterile pipette tips placed on the side. It takes a new tip for picking each colony, and then it places in these tubes. So if you see the image on the left, this is like a homogenizer to make finer particles of your molecule of interest. So if you are working on like biomolecules of interest, and if you want to make it in the finer particle or a slurry out of that, you need to pass through this. This is called a homogenizer. So coming towards, as Nanda Kishore was asking me, what exactly is agarogel electrophoresis and how it is detected. If you see this biorad machine here, this is called a gel documentation system. If you pull this pallet out, you place your gel over here, and then you just close this door. You have different protocols set in here, and you see, if you see here, you can see different bands. Those are seen in white color on this agarogel. Those bands, and you run a ladder or a marker at the extreme right or left of your gel to compare your bands of interest with your marker. So basically, this is an agarogel instrumentation. Now there is no need of this machine. Now you have a portable this gel documentation system, wherein you have a screen, and you can see your picture on the screen itself. And those are very costly. Those cost around $30,000 US dollar to $50,000 US dollars. So if you see the machine here, this is a laminar airflow to carry out all the aseptic conditions. So when you're working with tissue culture, you need to work in very sterile environment. There should not be any contamination of bacteria, fungi, or anything in your cell lines or your host of interest or your plant species. So what we do is, this is the surface where we work on. This is a UV lamp that is, once in the morning, this UV lamp is turned on so that before we start, all the microbes or contaminants present in the hood, they are destroyed using this UV lamp, or they are dead. And then we swap this with clean this cabinet with alcohol, and then we start working with our species. Like in our case, we work with plants, then we work with algae, and then we work with certain microorganisms. So to get rid of all airflow. So if you see, this is an autoclave machine. So to carry out all the lab operations, you need to have all your equipments, your media, everything well sterile. They are your equipments or your lab consumables, they should be free of all these contaminants like bacteria. If you use like contaminated conical flask or beakers, instead of growing your organism or host of interest, you may see some other bacteria or E. coli that is very commonly contaminant growing in your flask. So autoclave is a very much needed instrument in the lab. So this helps in autoclaving your media, autoclaving your lab instruments, autoclaving your lab consumables. So if you see, this is an orbital shaker wherein you can set your RPM, rotation RPM, to what RPM you want to rotate your flask, and this is the temperature range, what temperature range you want. And these are all closed cabinets, so you can keep them your flask rotating to have proper and uniform growth. And second thing, you can control the temperature as per as your requirement based on your host of interest. Suppose in case of E. coli, it needs like 37 degrees for its growth. In case of agrobacterium that is commonly used for plant transformation, you need 28 degrees for its growth and development. So these are like shakers, orbital shakers that are being used to raise cultures or grow cultures in the lab. Then coming towards, if you see the instrument here, this is called incubator, wherein you incubate your plates. at specified temperature, specified light conditions, so that you get a growth of your microorganism or your plant samples on these plates. So this the shapes and the sizes of these incubators vary. You have very big, huge incubators wherein we can keep around 100,000 samples or 10,000 samples of, or around suppose if you want to incubate small number of plates, you have a small number of small sizes incubators. So again, this is like that is called Kunar shaker for controlled experiments, wherein you don't want to get evaporation out of your flask. You want controlled humidity, you want controlled temperature, you want CO2 conditions monitored. So this is Kunar shaker, wherein you can grow your host of interest or species of interest or organism of interest at specific temperature, and then CO2 conditions, then humidity, and then light conditions. So what if you want 100 micro Einstein light, you can manage your light on the sensors. It's all sensor based, and you have a digital display in here. You can see what is the exact temperature inside the cabinet, what is the light inside intensity, what is the CO2 intake going inside the cabinet, and then what is the relative humidity inside. So based on that, you can fine tune all your growth conditions. If you see the picture at the bottom, this is a bioreactor. So once we develop everything at a flask level, then we go to bioreactor level, or that is called pilot 1 level, wherein we produce biomolecules of interest, our interest, in small sized bioreactor. This may range from 7 liters to around 100 liters. So in this we have exhaust inlets, outlets, then you have CO2 intake, CO2 inlets, O2 inlets, then you have inlets for media addition, then you have external then you have pH probes, then you have temperature knobs, and all those things fully automated. You can see all what is the pH inside the reactor, what is the temperature of your medium inside the reactor. Then you can also add media according to neutralize the pH or to adjust the pH. Then you have inlets for CO2, then you have inlets for nutrients like urea, ammonia, whatever you want to add, you can add from those inlets. And then you have outlets to take samples at different intervals for different analytical procedures or protocol to do some analysis on those samples to see whether you got the product of your interest with desired properties. So that's what I had, and now I'll be very happy if you have any questions regarding lab equipments, PCR, and any other general questions.
Yes, Dhokane sir. I think our principal sir has a question, but before that, I just wanted to bring one question. This one was actually from our professor from Chemical Engineering Department, Professor Vivek Kore sir. Are you there?
Yeah,
sir?
I can hear you.
Yeah, are you there, Vivek Kore?
Yeah, I'm here.
Yeah, so can you ask your question?
Actually, sir, I want to ask that can we use PCR machine for kinetic studies of reactions?
So kinetics in terms of what you mean, like a growth kinetics?
In general, because it has got, I think, temperature control and things like that, and we can keep small samples also. So that is why I thought, can it be used for kinetic studies?
Vivek sir, so far PCR machines are not been used for kinetic studies. So because in PCR machines, you can incubate samples for longer time. Suppose if you want to incubate something at 37 degrees, you can do that. But also you can range the temperature starting from, if you want to do something at 37 degrees for 2 hours, then 60 degrees for something at 2 hours. But you can't monitor growth because the sample size that goes in a PCR tubes is hardly around 100 microliters. And I think for kinetic studies, 100 microliter of volume will be too low to carry out or conduct or conclude anything out of that.
Okay, sir. Thank you. Thank you very much, sir.
Thank you, sir. And I just wanted to again mention something very different. So actually, audience मध्ये जे कोण मराठी म्हणजे ज्यांनी ऍक्च्युली जास्त कम्फर्टेबल मराठीमध्ये असतील, सर तर मला हे बोलायचं होतं की आपले आजचे स्पीकर जे आहेत डॉक्टर धनंजय ढोकळे हे ऍक्च्युली मराठी आहेत. हे आपले महाराष्ट्रातले आहेत, अहमदनगर जिल्ह्यातले, आणि तुम्ही त्यांच्याबरोबर मराठीमध्ये सुद्धा ऍक्च्युली संवाद साधू शकता. तुम्हाला जर मराठीत काय विचारायचं असेल प्रश्न तर ते अवश्य तुम्ही विचारा, ते मराठीत तुम्हाला उत्तर पण देऊ शकतील. सो जर तुम्हाला काय म्हणजे कम्फर्टेबल वाटत असेल की इंग्लिश मध्ये क्वेश्चन विचारण्यापैकी ऍक्च्युली जर मराठी जर ऑप्शन असेल तर तो आपल्याकडे उपलब्ध आहे. दुसरी गोष्ट मला सांगायची होती की सर ऍक्च्युली डॉक्टर धनंजय ढोकणे सरांचं ऍक्च्युली एक सगळ्यात आवडलेली गोष्ट म्हणजे काय तर त्यांनी कन्सिस्टंटली त्यांनी त्यांचं ऍक्च्युली बॅचलर डिग्री, मास्टर डिग्री आणि पीएचडी डिग्री मिळवलेली आहे. त्याच्यामध्ये त्यांच्या शिक्षणामध्ये बऱ्यापैकी कुठे गॅप नाही आहे. आणि त्यांच्याकडे ऑप्शन होता की त्यांनी ऍक्च्युली कॅनडाला जाऊन जेव्हा पीएचडी केले तर ते कॅनडामध्ये स्थायी होऊ शकले असते. तिथून ते ऍक्च्युली दुसऱ्या परदेशात गेले असते, पण ते आपल्या भारतातच परत आलेले आहेत आणि आपल्या भारतामध्ये ऍक्च्युली भारताला ऍक्च्युली त्यांनी सर्व्हिस देण्याचा ऍक्च्युली निश्चय घेतलेला आहे. सो राईट नाव ते ऍक्च्युली आपल्या रिलायन्स कंपनीच्या सिंथेटिक बायोलॉजी लॅब मध्ये ऍक्च्युली ते सायंटिस्ट म्हणून ऍक्च्युली काम करतात. सो मला ऍक्च्युली हा मुद्दा इथं आणायचा होता कारण तो सगळ्यात जास्त महत्वाचा असेल असं मला वाटतं माझ्या दृष्टीनं, आणि हे मला वाटतं मेन्शन करणं ऍक्च्युली जरुरी आहे. सो मला हे मेन्शन करायचं होतं. सो थँक्यू अगेन. मी ऍक्च्युली आय थिंक प्राचार्य सरांना बोलायचं आहे, तो प्राचार्य सरांकडे ऍक्च्युली ऑडिओ स्विच करतो. थँक्यू.
नाही, ओके ओके, थँक्यू. ऍक्च्युली
येस सर.
हॅलो डॉक्टर धनंजय सर.
या बोला सर.
Yeah. Hey, what I wanted to ask is regarding the copying. What is when you copy a gene, what is the material when we use for copying?
So, sir, what we use for copying is, so what we do is, if you want to extract or copy a gene from a genome, so first is we extract the whole genomic DNA.
Yeah.
So genomic DNA we extract, and then the genomic DNA has your genes of interest sitting on it. So suppose if I extract my genomic DNA, that genomic DNA will have around 40,000 genes sitting on that. Genes are nothing but it is a sequence of ATGC.
Yeah.
So what happens is, once I have the genomic DNA, then I design a signatures that will only amplify a gene that is of interest to me. Suppose if I want to amplify a gene from your genome, sir, and if I want to amplify a gene that gives black color to your hairs, so what I will do is I should know the sequence of a gene that gives black color to the hairs. Then what I do is I design a signature primers that will only bind to a gene that encodes for black color of our hairs. Once these primers sit on the gene of our interest, then using these enzymatic and temperature cycles, your double stranded DNA gets denatured, your primers they attach to your fragment of interest, and then you get your fragment copied to the length that the gene has, or your primers are designed. Basically, primers are designed at both the ends. So if you can see my video, if this is our fragment of interest, and you have one primer sitting in here, and you have one primer sitting from the other end. So this primer will run in forward direction, this primer will run in reverse direction. So you have complete double stranded your gene of interest copied. So once these signatures or primers they sit on your, see, this is your gene of interest and this is your primers which sits on your fragment. Then it starts putting nucleotides and it gets extended this way. It starts running in the forward direction. So you make copies. Any doubts, sir, or is that not clear yet?
I wanted to know just what is the material involved in it.
So that is a genomic DNA. Basically, so if you want to make a copy of your paper, Xerox copy of your paper, you need one initial paper to make copies, right?
Yeah.
So in that way, we have a gene sequence as our starting material that sits on our genomic DNA or DNA, and that DNA consists of, suppose for human genome, you have 40,000 genes sitting on entire of your genome. So, and you don't know which gene is at which location, which gene is at what location. So what you do is you design something that will just hit your gene of interest, sit on that gene and make copies, start putting your nucleotides and make multiple copies of that.
Okay. Thank you. And
yeah, sir.
you
Actually,
talked about
Yes, let me mention. So I think this is actually the little bit of a challenge, like because he is a biological biotechnologist and we are engineers, and you know they are actually the end users and the customers, and we are actually the developers, and like you know we are the creators for the equipments that they use. So this actually is a very actually soundful discussion that is happening here. Like you know if we can understand what their requirement is, like you know, so their requirement is from the biological point of view. And when he was mentioning about the material, I believe it is actually the biological material, which is, you know, so what he is actually adding into the PCR tube, that is actually kind of, you know, again, there is a big process to actually get that material. So there is a lot of like, you know, sampling technique, and then like, you know, sample preparation technique, and a lot of things are involved before actually getting that extract. Then he's actually adding that with other things, and then he puts in the PCR tube. So I think that is what that is correct, right, Dhokane sir?
Yeah. And Nanda Kishore, I will just simplify this in context to coronavirus. So once we collect swabs from throat or of the corona infected patients, what we do is we extract a DNA from those samples, and that DNA becomes our starting material.
Right.
And then you design those signatures or primers, those that will only sit on coronavirus gene, but those primers will not sit on human gene. So the primers designed will be very specific for coronavirus gene. Those primers won't be able to make multiple copies of a gene that come from human genome.
Yes, and I think to stress out, so the material is basically a protein, right? So if we actually go to the context, the material is a protein, right?
Protein, Nanda Kishore, the starting material is a DNA.
DNA is a protein, right?
DNA, you can say that, but the DNA is a starting material, and DNA codes for a protein.
Yeah, so basically it is a biological material. It is a biochemical material. Can we say that, like biochemical material?
Okay. Yeah, you can say that.
So, yes. So thank you actually, Dr. Dhokane, and thank you, Dr. Ghevde sir, for actually bringing this question. And this is what actually my point was, like, you know, can we actually have a discussion with biologists? Can we have actually a discussion with chemists? Because what we think is, like, you know, we are engineers, and like, you know, we always think from the industrial point of view. But right now the industry is actually very complex, and we should actually always start to have dialogue with, like, you know, people who exist in different actually sections of the industry, and R&D is actually one of the section. So so now actually I want to ask some questions, like, you know, so is there any question from the actual, like, you know, instrumentation department? So Karan sir, please. Thank you.
Okay, my question is, which are the different types of the diseases so that we can going to be used by using the PCR? And another one is whether the PCR is used to prepare the medicines on that particular diseases.
So, sir, all the genetic diseases, those are linked with your DNA or gene sequence, can be identified using PCR technique. Suppose now I will give a very simple example. Suppose a gene that cause like sickle cell anemia, a disease known as sickle cell anemia. That disease is encoded by a gene. So now if you see a healthy patient and a disease patient, you amplify or make copies of this gene, get this gene sequence from a disease patient and a normal patient, and then you compare the sequence data from both the patient If you see some variation in the sequence from normal patient in comparison with the disease patient, that means you have some sort of mutation, you have some sort of deletion, you have there is some change in your gene of interest, so that you got disease.
And another question, Karadi sir, you had two questions, right? What is the second? Yeah. So how can it be used for actually drug discovery? That was the question, I think.
Yeah. Yes.
So in terms of drug discovery, so now you know this, you need to have some sort of a drug that interacts with some sort of a ligand, right? So in terms of drug discovery, we find the functional domains of your gene. So in technical terms, that is called a domain or active site that is present in the gene. Then we designed some sort of signatures. When those signatures amplify in a patient, then only that will be used. Otherwise, that won't be used. This is how that will be employed in drug discovery.
Okay.
There is one question from the Yahoo chat. It is actually from one of our IT departments, Professor Mr. Pvpit. Does PCR machines are used in cyber forensic labs? If yes, then how is it useful?
Yes, PCR machines are being used in cyber labs, because if you see these CID serials and all those things, you see they collect DNA samples. And if you see my DNA sample, your DNA sample, and everybody's DNA samples, everybody has different DNA composition. So if somebody is a victim or somebody has done something wrong, then you collect the sample and you take the samples from both the places. Suppose if you find some blood samples which has been split at the site of incident, and then you collect the sample from the suspect, and then you do this PCR analysis and do sequencing, and if both the sequence analysis, if they match, that means the guy who was present at the site is the one who has done the crime. This is how they compare. Basically what they do is they amplify the genes from both the samples, and then they do sequence analysis, and then they come to the conclusion.
Okay, thank you. I hope Mr. Falle sir, Professor Falle, you actually got the answer. Is there any other questions from Institute department?
Hello.
Yes, who's it?
Hello.
Yes.
Yeah.
Yes.
Yes, sir.
Sagar Sultan,
So,
please.
sir, yes, sir. Yes. So my question is, what are the different sensors are present in PCR machine?
So basically in PCR machine, you have only temperature sensors. You don't have any other sensor than temperature, because here only we need an enzyme that works at different temperatures. So from 4 degrees to 99 degrees Celsius, the machine works. And once we set a protocol saying that in first step, we need 95 degrees Celsius. In second step, we need 75 degrees Celsius. In third step, we need 55 degrees Celsius. And once the PCR reaction is done, we need to incubate our samples at 4 degrees Celsius, so that during that storage time, our DNA or your PCR product, it won't get degraded. So basically only this machine has a temperature sensor.
Okay, sir. My second question is any calibration needed for PCR instrument?
So in terms of calibration, what we do is we just do temperature calibrations. See, in all the wells, you get uniform temperature during each cycle. So these vendors, they come back and we have this annual maintenance contracts with them. So they every 6 months or every once in a year, they come and they check all the wells. Sometimes what happens is you keep the same samples in all 8 wells. In some wells it amplifies, in some wells it doesn't amplify. In this case, there may be 2 reasons. Sometimes when you prepare this mix for a PCR reaction, something you might forget to add in one a tube. But what we do is we make a PCR mix together, then we suspend in all the tubes and we keep it. But sometimes what we have seen is in well 1 it works, well 2 it doesn't work. This may be
Okay.
because the temperature range, whatever is needed, it doesn't achieve that range. So
Okay.
once in 6 months you need to do these temperature calibrations.
Okay, sir. Thank you.
Okay, so
Thank you very much. Thank you.
anyone else has a question? I have one more question. When you mentioned about the price of the different PCR instruments, you mentioned 25,000 to 2.5 lakhs. So that was in US dollars, right? 25,000 US dollar to 2.5 lakh US dollar.
Yeah,
Or is it like 25,000 Indian rupees to 2.5 lakh Indian rupees?
So some machines are like you can even get in 25,000 Indian rupees.
Okay.
So wherein you can just plate 8 samples or 48 samples, and you can get a handy machine with 384 wells around 2.5 lakhs
And I
in
have one
Indian
more
currency.
question. Yeah, and I have one more question. Basically, like in this chemical and biological labs, I just wanted to bring this in front of everyone, because in this chemical and biological labs, what they use is called something a pipette, right? A pipette. So
Yeah.
in many students from science students in India, what they actually have, they have a glass pipette. So what you do is you suck the fluid, and then you actually suck it till the bulb portion actually exists, and then you quickly actually hold the top portion with the finger, and then you actually that pipette actually has a graduated scale, and then you actually with your finger you actually release the fluid up to the level we want to actually achieve the quantity, and then eventually what you do is you again transport that to the sample where we actually want to do something with that, and then you actually leave your finger, so it will actually go by gravity, and then you blow the last drop out, right, which is the manual technique. So in US and Canada and even in India, definitely there are actually different kinds of pipettes, right? So they are actually mechanically controlled, the volumes can be controlled with some kind of a rotating screw, and they are like very spring loaded ones, and then there are also like some coming in the digital form, so it can also tell the exact volume in what you said 100 micromilliliters, something like that. So do you know actually I mean what are the different makers of those kind of pipettes, or if you have some kind of a picture you can share on the screen right now, what is a pipette, and so maybe if we can actually probably build our own pipettes in India, and and maybe the front actually the tips of the pipettes, maybe something like that, and actually do they actually manufacture that in India, or actually it is being manufactured outside India? So if you can actually probably discuss something on that, if you are actually okay.
Yeah, Nanda Kishore, I think pipette is a very important part of a molecular biology lab. So if you have seen our old 11th, 12th days when we have these titration experiments, as you clearly said, we used to blow the liquid using those glass pipettes. Those glass.
Sir, कोणाचा तर pointer actually. Okay, carry on. The sound is gone.
So as you clearly mentioned, initially we had those like glass pipettes, wherein we used to suck in the liquid and then used to drop it out into whatever volume we wanted. But nowadays, in all the molecular biology labs or screening labs, you have these pipettes wherein you can aspirate the required volume. So it starts from 0.5 microliters to even we have pipettes around 10 ml. So and the Ipendorf is a company that makes these pipettes, and I don't know, I'm not sure whether this is, if they have any of their manufacturing unit in India, but Ipendorf is one of the leading companies that provide these pipettes and tips to all the molecular biology labs. So what these pipettes how they are used is you can aspirate known volume of your interest, and then you can put it in any solution for solution preparation or making different chemical combinations. So pipettes, you just aspirate it with your thumb. You just put your hold your pipette, press your thumb, and you get your liquid into your pipette tip, and then you put it wherever you want. And if I can show it on the internet, I can do that. Just a minute. But if I don't know whether if I need to share the screen again.
I think you have to start your browser, and then you have to again stop sharing the screen, and then you have to again start sharing, and then you share the browser from the selection menu.
In between, if you have any question, I am.
Yeah, in between. So, anyone else has any questions? There are a lot of faculty participants in this group. So, any other faculty participants in this group, if they have any questions, or any other YouTube viewers, if they have any questions, please go ahead. Yes, More sir, please.
Sir, I want to ask this question. Means, sir, may I ask?
Yeah, go ahead, sir.
Yes, sir.
Sir, this PCR machine is used to find out the infection disease. Is it useful now for this corona infection? And how much time it will take to find out the, or it will give the result after the taking sample?
So, all the detections, sir, now for coronavirus are being carried out using PCR machine. So the turnout time is around 2.5 hours. So once you get your samples, one of our unit from Reliance is also doing sample diagnosis for coronavirus. So initially you need time for labeling and putting all the samples together in the right place and then labeling them. Once these samples is received in the lab, it takes around 2.5 hours to carry out a PCR reaction on your swab samples, and then the detection takes around another 2 hours. So around in 4.5 hours, you can get your PCR, your coronavirus test, whether you are positive or negative. And now Reliance is also doing this kind of screening for patients in Mumbai. And if today morning, if they get patients, next day morning, they submit the reports back.
Okay, sir. Okay, thank you.
Yes, so and then the question here is like, so because the PCR instrument contains actually, like, you said like 96, 48, 348, like, kind of wells. So it means that those many samples can be placed at one time. So do they actually keep only one sample always, or is there always a redundancy, like, two samples, just one as a backup, like, to confirm if there is a false positive or a false negative, actually kind of a trend. So do they actually have a redundancy, or they just put like one sample and that's it? And so like 348, so 348 samples from 348 suspects. So what is the standard?
So Nandakishore, the accuracy of PCR is like, I would say 95%. So once you do your PCR reaction, if you have like 100 samples ready with you for a PCR reaction, in one go you can keep 94 samples if you have a 94 well machine. If you have a 384 well machine, you can keep 384 samples in one go. There is no need to repeat your samples. But in case of coronavirus, as this is very difficult, I would say, times for people, if they come to know ki even if they are not coronavirus affected, and if somebody tells them you're positive, they will be really afraid about this. So what is being done here is the same sample, two guys are doing PCR reaction on the same samples to have a cross confirmation. So once the PCR is done by two different individuals, and when the same results come from both the individuals, then only it is being told, or it is being communicated whether patient is positive or negative.
Okay, thank you so much. This is actually very very clear confirmation actually from your end.
So if you see the pipette picture, you can see on the screen.
Yes, we can see it.
So basically, this is a pipette. Using your thumb, you just push this knob down, And if you see, this is a pipette tip wherein you can aspirate the required volume. If you need, suppose 100 microliters, 200 microliters, 500 microliters, you just press it down with your thumb, you aspirate it and put it in your PCR tube, other pen drop tubes, or 10 ml tubes or 20 ml tubes. So if you see, the cost of this is like $30. And just a minute now, and this is the company that is the world leader in providing pipettes. So this is the set of pipette. So now here, this is like 2.5 microliter pipette, this is 10 microliter pipette, this is 20 microliter pipette, wherein you can aspirate 20 microliter, this is 100 microliter, 200, and this is 5 ml. So now here you can aspirate a volume using a single tip once at a time. But if you say multi-channel, you have a pipette wherein you can use 8 tips at a time, and you can aspirate solutions from 8 different tubes. So these are called multi-channel pipettes, and this also comes in varying range from 0.5 microliters to 10 ml.
Okay, thank you, Dr. Dhokane. So, dear instrumentation students and faculties, so this is very important, right? So, the pipette is actually the most important part of any kind of a scientific instrument, scientific experiment, and because what you put is it, if it is going to be in the correct, then only you'll get something output in the correct. So the volume of the sample which is going to be tested, so that has to be very very precise. And can you actually Dr. Dhokane sir, can you explain how do we calibrate this pipette? So because it needs to be confirmed that okay, what we are doing it is going to be precise. So they actually calibrate it, and then they do it in mass, and then they have to do some sort of calibration, right? And if the calibration is wrong, then there is some kind of a rotating knob that you have to rotate, and then you set it, right? So if you can just give a little bit talk of that, how do they use it? How do they do calibration using a weighing scale method, or if there is any other scale method?
Yeah, calibration is being done using weighing scale method. So if you have like 200 microliter pipette, you just aspirate the volume, and then put that volume in small measuring cylinder or a beaker, and then you put that beaker on a measuring scale. And if you see that it comes to the same range, that means your pipetting or your pipette is good to go, and you need not to have calibrations. But if your the liquid calibrations, if they fall away from whatever volume if you have aspirated, and when you put it on scale, if it deviates very much from the range that it should give on the scale, that means you need to give your pipettes for calibration. And again, the guys from Drop, they come once in 6 months, and then they calibrate with those pipettes using distilled water.
All right, thank you. Is there any other question from any other end? Or should we actually start the conclusion of the session? मराठीमध्ये कोणाला प्रश्न असतील तर अगदी बिनधास्तपणे विचारू शकताय. सर आपले मराठीच आहेत. हिंदी में भी अगर किसी को क्वेश्चन है तो हिंदी में भी बात कर सकते हैं. कोई है जो एक्चुअली दूसरे एंड पे हिंदी में बात करने वाले में कंफर्टेबल है, तो सर कैन ऑल्सो आंसर द क्वेश्चन इन हिंदी. If there is not anyone who is interested to actually ask any question, or who is not available to ask any question, maybe we can start the conclusion. Sir, is Institute Department Karate sir available?
Nandakishore, just before ending the session,
yes,
if anybody or of your students, they want to get in touch with me, you can provide my email address, my phone number, WhatsApp number, so that if they have any doubt, they can connect to me in person. And if they need any help in seeking some sort of internship at these premier institutes like Drop, BioRad, Thermo Fisher, I can help them out in getting those things.
Yes, sir. Thank you so much for mentioning about this. And definitely there could be a lot of students who might be contacting you very soon on this aspect. And is our principal actually, dear Dr. Ghede sir, are you actually interested to probably speak on something in terms of actually relation?
Yeah, that is what he rightly mentioned. Na? We
Yes.
can have some internships through Dr. Dhanajai sir, and
Yes, sir.
we can also invite him for some lectures. Already, see, we are facing some problems with the applied biology subject we have. I think in
Yes.
second year
Yes.
instrumentation. We
Yes.
can have some guest lectures from Dr. Dhanajai sir
Yes.
so that the students will get an idea about that subject and they will learn very nicely. And then we can have uh, interactions with him. We will always try to uh, call him for some or the other purpose. As now we have developed some relationship with him. You know him personally, so that we can definitely call him back, and
Yes, sir.
we'll discuss some of the issues wherein we can have his involvement. How can he mentor us for some of the issues related to the development of instrumentation department? Huh? So you
Yes,
can
sir.
utilize his expertise. Uh,
And
and
to all the students of like instrumentation department, I would like to bring to your notice in this field you have a lot of opportunities. All the molecular diagnostic labs, molecular biology labs, they need like huge number of equipments. Those are very costly. We have some instruments which cost like 1 crore in Indian currency. Those are robotic systems, and you have a lot of opportunity to work on this system, modify this system, automate these systems, and try to bring systems that are user friendly, tabletop, which can be used in a small room and can solve all your purposes. So I think you have very good career opportunities. If you I will also share my presentation with Nandakishore, and then you can go through the links of all those companies and try to see what these companies they manufacture and where you can get in or put in your expertise there, and how you can bring in some instrumentation, or I would say automation in this kind of instrument, and try to bring some newer sort or like instruments to the market.
Yes, sir. Thank you so much. Also thank you so much on the behalf of actually students of instrumentation department. And now sir, Dr. Dhokane sir, do you want to say anything, or should we start the conclusion?
Yeah, go ahead, Nandakishore.
Okay. Mr. Karade sir, Professor Karade sir, are you available? Karade
Hello.
sir?
Yes, I will.
Yes. So should we start the concluding of the session? Hello? Sorry, in the background, actually, I have my kids and they are actually not leaving me. So maybe there could be some background coming. Excuse me for that.
Hello, sir. Who is going to conclude the session?
Actually, Karade sir, probably he lost the communication.
Yes,
Just a moment, he's actually calling me. Priyanka Patil, madam, एक मिनट था वहाँ सर बोला. तुमसे network ये लगा?
Let Priyanka Patil madam, if she wants to conclude, let her conclude. Otherwise,
okay. Madam Patil, na? Patil.
Yes, sir.
Yes, Priyanka Patil madam. So Karade sir actually is network is not actually doing great. So you can. So he just mentioned that you can start the conclusion.
Okay, sir. Okay. Good afternoon, all. I am Priyanka Patil, Assistant Professor PVPIT Budhgaon. It gives me great honor and privilege to propose vote of thanks on this SDP. I am grateful to our guest, Dr. Dhanajai Dhokane sir, who despite his busy schedule and had time to grace this occasion. Thank you, sir, for sharing your experiences, views, and all the knowledgeable talk. Once again, I thank you on behalf of PVPIT family and also from instrumentation department. Once again, I thank you all the guest speaker of this SDP who share all the knowledgeable things with our students and participants. Above all, I thank you all students, participants, as your participation had made this session successful event. I also thank you all faculty members, HODs of all department of PVPIT, Dean Academics, Dean R&D, Arugre sir, Dean Academics Kumbhoskar sir. I also thank you, Dr. Dhole sir, to give us such a good speaker for this SDP and made this SDP grand success. I also thank you, our HOD, Sir Karade sir, and all organizing committee members. Last but not least, I also thank you, our principal, Dr. D.V. Ghevde sir, Honorable Trustee Amit Dada, and Chairman Honorable Vishal Dada Patil for providing support for this SDP. Thank you all.
Yes, thank you all. Apologizes if there was any delay in the session, actually due to technical glitch, and we are all learning. Many actually things from doing such webinars, and I think we are pretty much ready for accepting the challenges of actually conducting or hosting any sessions. And thank you so much, everyone. Okay, so thank you so much, Dr. Dhokane, and I'll again get back to you later. Thank you so much.
Thank you so much all.